In this lecture, we're going to explore the structure of eukaryotic cells. And later, we'll talk in another chapter about prokaryotic cells. So first of all, ask yourself, what's the difference between these two cell types? And we'll come back and answer that in a few minutes. Cells range in size, and some you can see without a microscope with the unaided eye. So you can see here, for example, that a lot of eggs you can see, like we know that we can see the chicken egg here. With our naked eye, frog eggs, if you look carefully in a pond, you might see a cluster of these. And if you knew you were looking for a human egg on a slide, you might be able to see it here, around 100 micrometers. But most cells, most eukaryotic cells, are in this range here between 10 and 100 micrometers. So you want to know this. You want to remember this size of eukaryotic cells. Another thing you want to remember that we'll come back and talk about later, but is that prokaryotic cells are smaller. They range from about 1 to 10 micrometers in general. So something else you want to remember. So that is one difference between prokaryotic and eukaryotic cells, is their size. So with the eukaryotic cells in general being 10 to 100 times larger than the prokaryotic cells. So because of their small size, most cells require a microscope to view them. So over here on the right, you can see that LM stands for a light microscope, and you need a light microscope to view most cells. Now you can also view them with an electron microscope, but we'll see when you do that, you can see much more detail inside the cell. You can look at the structures within the cell and see them, whereas with the light microscope, a lot of times you can't see those same details. So it wasn't until microscopes were invented that we really began to understand that organisms are made up of cells, that this is kind of the building block of all organisms. And Anthony van Leeuwenhoek was the first one to make a lens that would magnify an object. And here's his microscope up here at the top. A very simple microscope, and he put his little lens that he polished, this biconcave lens, in the center of a slab of wood, and then he'd put his specimen on the end of this pointed probe and move it in front of his lens and hold that up to his eye. And then later, in that same century, the 1600s, other microscopes were invented, and these were more stable, and you could see the specimen easier. The lenses got more powerful. So down here on the bottom is a microscope that was invented by Robert Hooke. Sorry. Okay, there we go. And later we got even more complex, so that now we have microscopes with multiple magnifying objectives, like we'll use in class. And you can see here that the organisms that we work with have also gotten very smart and know that they're in trouble when they see a giant eye looking at them. I like that comic. But the work at improving microscopes and using them to look at plants and animals over the next 200 years, from the 1600s to the 1800s, led to what we call the development of a theory, the cell theory, that states the following, listed here, that all organisms are made of cells, and the cell is the simplest collection of matter that can be alive, that cells are related to other cells. They don't just appear from nowhere, that they come from earlier cells. and that cells, they can differ a lot in their characteristics and sizes, for example, as we showed in the last slide, but they share a lot of common features. So these are all components of the cell theory, and this is something you want to commit to memory. What are the different components of this theory? And then with the invention of electron microscopes in the 1950s, we've been able to see inside the cell at the ultrastructure of the cell, and we'll see that means that we can learn a lot about the different organelles. And there's actually two different types of electron microscopes. There's transmission electron scopes, where you can see through a specimen, and that's shown at the top in A here. So this requires very thin slices of your specimen and an electron beam to illuminate it. And this is helpful because the electrons, the wavelength what they move at is much smaller than the wavelength of photons in light. And so we can resolve 2 points that are closer together with electron microscopes than with light microscopes. So for example, light microscopes can resolve down to about 2 points that are about 0.2 micrometers apart. So this is a light microscope. Whereas with, for example, the transmission microscope, it's about 1,000 times better. We can get down to 2 points. Sorry, my implement's not working very well here. That are about 0.2 nanometers apart. So 1,000 times smaller. And then with the scanning, it's not quite as good. This is what we're doing is we're looking at we're scanning beams of electrons across the outside of a specimen to see more of the 3D structure of it. And that's about, you can see points that are about 10 nanometers apart. So that means, say, with light microscopes that we can get up to magnifying objects about 2,000 times. And with the transmission electron microscopes, we can get up to about 100,000 times magnification. And with the scanning electron microscopes, about 10,000 times. So these electron microscopes are much more powerful, but it's a lot harder to prepare a specimen for them as well. All cells, whether they're prokaryotic or eukaryotic, have a plasma membrane that surrounds the cell and controls what goes in and out. And within the cell, they have what's called the cytoplasm, and that's basically everything inside, the semi-fluid medium that's inside the cell and the organelles that are suspended in that. And now, all cells have a cytoplasm, right, but not all cells have organelles, because we did talk about this is another big difference between the prokaryotic and the eukaryotic cell, is that the prokaryotes don't have the organelles. So in the rest of this chapter, we're just going to be talking about eukaryotic cells with these organelles. And then the definition of the cytosol, which sometimes you see those two words, cytoplasm and cytosol, used interchangeably, but the cytosol really just refers to that semi-fluid medium, but not everything inside the cell. Okay, so eukaryotic cells have the organelles inside, these compartments that separate different functions within the cell so that different types of reactions can occur in different locations. And these organelles are surrounded by membrane that's very similar to the plasma membrane. And the organelles of eukaryotic cells, animal, plants, fungi, are all very similar. There are some differences, though, particularly between animal and plant cells. And down here we see that animal cells that these two organelles, I guess I should say, centrioles and lysosomes, are only found in animal cells, and chloroplasts are only found in plant cells. And we'll talk about that again. Okay, so here's a figure of an animal cell, and it shows the location of of the major organelles. And we're going to talk about each one separately. So at this point, I'm not going to talk about or point out individual organelles, except to say, like, the nucleus here is often towards the center of the cell, but not always, depending on the cell type. So here's our nucleus, and the other organelles are arranged around it. So you want to be able to identify and point out different organelles in a picture like this. And here's a similar picture of a plant cell. Now these are cells where the nucleus often gets kind of pushed off to the side because they have a large central vacuole filled with water, which takes up much of the center of the cell. And remember, plants, again, are the only cells that have chloroplasts. Well, there are some protists, some algae cells that also do. But here's the chloroplasts in this plant cell. Another thing that plant cells have that animal cells don't have is a cell wall around the plasma membrane. So there's the cell wall of this plant cell. So again, just like with the animal cell, you want to be able to identify the different organelles in a plant cell and know the difference between this and an animal cell. The rest of the lectures from this chapter, chapter 6, focus on the different groups of organelles in the eukaryotic cell. And this table at the very back of the chapter is a nice summary of at least a couple of those groups. So you can see they've got the nucleus up here at the top, and then the endomembrane system as the second group. And together you can kind of consider those part of all the endomembrane organelles, as we talked about the nuclear envelope at least being part of that system. And then, so that's like one group of organelles. Another group are the energy-related organelles that we talked about, the mitochondria and the chloroplast. And another one, the peroxisome, that's specialized in breaking down metabolic waste products. And then the third group of organelles is not on this review table, but we talked about the cytoskeleton. So I'll make that our third group here. That as one that you should remember as well. So, and you want to remember the three components of the cytoskeleton. Remember the microtubules, the microfilaments, and the intermediate filaments, and their role in cell shape and form. As all three of those play a role in that. And then also the microtubules and the microfilaments, we talked about their role in any kind of movement in the cell or of a cell or organism. So this is a nice way to summarize the material in the chapter. And this table, again, gives, the name of the organelle at the top, and then what its structure is, so you want to know that for each organelle, and what its basic function is. So that's what you really want to concentrate on learning in this chapter.